As I said, I'll be taking a series of about 3-4 lectures on plant transformation strategies. This is lecture 1. This is the usual disclaimer that this is meant for you people only, particularly School of Life Sciences of KBC and EMU during this lockdown due to COVID-19. And it should not be used for any other purpose, copied or posted on any site without the permission of author, that is me. And some of the pictures in the presentation have been taken from the public domain websites to make the topic easy to understand, and the credit for such pictures remains with the original publisher. Alright, so the question is, what is the history of crop improvement? Basically, as I told you earlier, that this plant transformation strategies, we are understanding from the point of view of their role in crop improvement. Okay, so what is the history of crop improvement? So broadly speaking, by trial and error, we have been doing this crop improvement for the last almost 10,000 years. Last almost 10,000 years. By applying scientific principles of breeding, so you understand breeding is a branch of genetics. So by using those principles of breeding, we are doing this for last almost 100 years. And using chemical mutagens, chemical induced mutation, for last almost 75 years, we are attempting this plant transformation, or means transformation of desirable traits into the crop plants. And by using rDNA technology, recombinant DNA technology, we are using it for last almost 40 years. We started using recombinant DNA technology somewhere around 80s. So it is almost 40 years now that we are using this recombinant DNA technology for transforming the plants with the desirable genes. Right? So this is broadly the history of crop improvement. Now, improvement in crop, as I said earlier, we have been doing it for last almost 10,000 years. And plant breeding later on became a very sophisticated branch of applied genetics. And you will have to agree that our breeders have developed elegant strategies for crossing plants to introduce and maintain desirable traits. So, and this has resulted in what? This has resulted in improvement in a number of economically important crops. So whatever crops, economically important crops you see around presently, for example, wheat, rice, these are as a result of classical breeding experiments, because their yield has increased tremendously, and as a result of that, our country can feed the ever-increasing population. So we have those kinds of breeds available of economically important plants, and this is primarily the work that has been done by the breeders. Now, if it is so, this plant breeding is has played such an important role, what is the need? See, basically what happens, classical plant breeding is very slow and uncertain. Okay, so you have to do experiments, it takes time, and after the time is over, you really don't know whether you are successful in crossing over the desirable trait or not. Second bottleneck is sexual compatibility between the plants. The two plants where crossing taking place, sexual compatibility is necessary in classical plant breeding. And genes, in addition to desired genes, they also may get transferred. Okay, because it is uncertain, it is not in your hand. Okay, it is uncontrolled. So besides the gene of your interest, it may so happen that additional genes, undesired genes, may also get transferred. So these are the three major bottlenecks of classical plant breeding techniques. One is that it is slow, time consuming and uncertain. Second is sexual compatibility is necessary. And the third is that genes in addition to the desired genes may also get transferred. But in case of rDNA, recombinant DNA technology, all these limitations, they can be circumvented. You can bypass all these limitations. So what does it mean? It means that it is fast, it is certain, it is sure. You can do transformation, and there is no pre-requirement that the plant that is being transformed should be sexually compatible, and you are sure that the gene of your interest only is getting transferred. Now, what are the different strategies they are available for plant transformation? So there are quite a few now, but the two major one is agrobacterium mediated transformation, and the second one is microprojectile bombardment using a shotgun. These two are the most common. In fact, of these two, agrobacterium mediated transformation, this is the oldest one, the first one, and later on, this was invented, microprojectile bombardment. But in addition to these two primary strategies, we have additional strategies. Viruses also can be used as a vector for plant transformation, and there is one more technique called electroporation. That also can be used for transforming the plant cell with the DNA, with the foreign DNA. Okay, but these two more or less are of academic interest. These two, the first two, they are being commercially exploited, they are being commercially used. So let us start with the agrobacterium mediated transformation. Now, in the first place, the question that comes is, what is this agrobacterium? So agrobacterium is a soil bacterium, and it is closely related to rhizobium. So all of you must have heard this name, heard this word, rhizobium. So this is a close relative of rhizobium. It resides in soil, and therefore it is also called a soil bacterium. And it causes crown gall disease in plants, particularly in dicots. So you have primarily two major categories of plants. You have dicotyledonous plants and you have monocots or monocotyledonous plants. So this agrobacterium, it causes crown gall disease in dicots. Okay, and what is this crown gall? It is shown over here. This is crown gall. It's a tumor-like structure. It's a tumor-like outgrowth, primarily on the stems of these dicotyledonous plants. Okay, so this disease, this gall formation, this tumor formation is a result of agrobacterium infection. This bacterium, it contains a large plasmid. It is called as TI plasmid. TI का मतलब होता है tumor inducing, because it induces tumor formation. Therefore, the plasmid is called as TI plasmid. TI, tumor inducing. And the bacterium per se, though it is a bacterium, it's a complex bacterium. The genome has been fully sequenced, and it has got 4 chromosomes and close to 5,500 genes. So for a bacteria, this is too many genes. Okay, and therefore we have used the word, it's a complex bacterium. Now, this particular bacterium, it can survive in the soil independent of plant host. It can survive even without the plant host in the soil. It can remain in the soil in a viable condition. It infects plants through breaks or wounds. So wherever there is an incision, there is a wound, there is a cut in the plant, that is the site of infection. This bacterium invades the plant tissues through those wounds, through those breaks. And it is responsible for that common disease of woody shrubs, herbaceous plants, particularly problematic to members of family Rosaceae. Okay, it causes crown gall in those plants. Wherever those plants, they incur an incision, a wound, a cut, that becomes the site of infection. And once it infects the plant, it causes crown gall formation, it causes tumor formation. The tissues, they bulge out, It's an outgrowth of the tissue, uncontrolled growth of the tissue at the site of infection, resulting in the formation of the crown gall. And these galls are spherical wart-like structure I have shown you in the previous slide, similar to tumors. So technically it is called as crown galls, but you can also use the word tumor. Now this is the host range of this particular bacterium. Host range of this particular bacterium is, about 331 genera and 641 species it can infect. So we call it as a host range of this particular bacterium. Monocots generally, usually, they are not infected. Except some members of family Liliaceae, they are not susceptible. Monocots are, as a general rule, they are not infected. They are not affected by this particular bacterium except some members of family Liliaceae. So, as I said earlier, infection occurs at the wound site and it involves recognition and chemotaxis of the bacterium towards the wound itself. So you can understand, you can visualize also that wherever there is a wound, wherever there is an incision, wherever there is a cut, okay, from the wound side, some chemicals are released and these bacteria are attracted towards the wound by chemotaxis. Okay, and they will infect the plant tissue. Now let us understand something about the TI plasmid, the tumor-inducing plasmid, which these plants, which this bacteria contain. It's a large plasmid. I told you 200 kb. 200 kb for a plasmid is a really big size. Circular plasmid, the size is around 200 kb. So pretty big plasmid. It contains genes for virulence. Genes for virulence means it contains genes which are required for successful infection. Okay, so all those genes, the proteins of which, these gene products of which are required for infection, they are there residing on this TI plasmid. Besides virulence, genes for virulence, it also contains genes for host-directed opine synthesis and catabolism. So opines are a group of amino acids. Okay, so host-directed. The genes are there in the bacterium, but these genes are expressed only in the plant cell. Once the bacterium has infected the plant cell, then only these genes are expressed, and therefore we are using the word host-directed opine synthesis. Opine, as I said, are a group of amino acids, so their synthesis and their catabolism also. Okay, so they will be synthesized in the plant cell using the plant machinery, using the plant resources. Okay, and they will be exclusively utilized by the bacteria for its growth and development. Besides the opine synthesis, genes for opine synthesis, this TI plasmid also contains genes for host-directed bacterial type plant hormone synthesis. Okay, again host-directed. That means it contains, the bacterial TI plasmid contains genes for auxin and cytokinin, but those genes are not expressed. Those genes are expressed only when the bacteria has infected the plant tissue, and therefore we say that they are host-directed bacterial type plant hormone synthesis, genes for host-directed bacterial type plant hormone synthesis. The genes for opine and plant hormone synthesis, they are contained on the tDNA. So this tDNA is a part of TI plasmid. So I'll be showing you a picture of the TI plasmid, and then you can understand. This tDNA is a portion of the TI plasmid, and this tDNA, the portion of TI plasmid we are referring to as tDNA, it contains genes for opine synthesis and genes for plant hormone synthesis. These two types of genes, they are clustered, they are grouped, and they are located on that portion of DNA we are referring to as tDNA. All right. Now this TI plasmid, it can stably integrates randomly into the plant genome. So after infection, this TI plasmid gets stably integrated into the plant genome. Please remember, the site of integration is not pre-decided. The site of integration is not predetermined, and therefore we are using the word randomly. So there is no pre-decided, predetermined sites of integration of TI plasmid into the host genome. It integrates itself randomly. Okay. And expression of genes in wild type DNA, tDNA, and I just told you that this tDNA contains genes for opine synthesis and genes for plant hormone synthesis. So when these genes get expressed, locally you have large quantities of plant hormones in that localized area. Localized area means the site of infection, where there is a wound, that is the site of infection. So in that particular area there is a too much concentration of plant hormone, resulting in an uncontrolled growth of tissues over there, resulting in the formation of tumor. So expression of genes in tDNA results in dramatic physiological changes in the plant cell. What is meant by this dramatic physiological change? Dramatic physiological change means, since you have too much concentration of these plant hormones in that localized area, that results in uncontrolled growth of the plant tissue, leading to formation of crown gall, leading to formation of what is called as tumor. Synthesis of plant growth hormones, as I said earlier, auxin and cytokinase, leads to neoplastic growth, uncontrolled growth. Technically it is called as neoplastic growth, leading to tumor formation. Okay. Now, as I said, these bacteria, once they infect the plant cell, they synthesize unique amino acids. Together they are called as opines. So there are primarily three different types of opines. You have what are called as octopine or octopine, nopalin, and agropine. Of these three types of unusual characteristic amino acids, unique amino acids, octopine and nopalin, they are derived from amino acid arginine. They are derivatives of amino acid arginine. Whereas agropine, it is a derivative of amino acid glutamate. Okay, so please remember, I am repeating that once it infects, it carries along with it on its genome, on its plasmid, TI plasmid. In that TI plasmid also there is a portion of TI plasmid that is called as tDNA. It carries genes for opine synthesis. Okay, these genes are there in the bacterial plasmid, but they get expressed only in the plant cell because they are controlled by eukaryotic regulatory sequences. So unless they infect the plant cell, those genes will not get expressed. And there are basically three types of opines. These are unique amino acids. You have octopine and nopalin, which are derived from amino acid arginine, and you have agropine, which is derived from amino acid glutamate. Now, type of opine synthesized depends on the strain of the Agrobacterium tumefaciens. Okay, not every Agrobacterium will synthesize all these three types of opines. They will be synthesizing only one type of opine, and the type of opine synthesized depends on the strain of Agrobacterium tumefaciens. That means it will be carrying only that particular opine synthesis gene. Opines are catabolized by the bacteria, which can use only the specific opine that it causes plant to produce. So don't go by the complexity of the sentence. What does it mean? It means that the opines being synthesized in the plant cell using the plant machinery, using the plant resources, are exclusively utilized by the bacteria for its growth and development. Okay, and plant cannot utilize this opine. These opines will be utilized only by the bacterial cell. Okay, and this catabolism by the bacteria, uh, it catabolizes only that opine which it causes the plant cell to synthesize. Okay, only the specific opine that it causes the plant to produce, right? So this is the diagram of the TI plasmid. So this particular region, from here to here, this region, it is called as tDNA. This entire plasmid is TI plasmid. It's a big circular plasmid. Total, size is around 200 kb. Of that, a portion of DNA of the plasmid is called as tDNA. What does it do? It carries genes for plant hormone synthesis, means genes for auxin, genes for cytokinin. This is the gene for cytokinin. This is the gene for auxin, and it also carries the gene for opine synthesis. Okay, and this tDNA, it is flanked by 225 base pair direct repeats, which are called as right border. This is right border, and there is a left border. This is left border. These are 25 base pair direct repeats. Left border on the left hand side and right border on the right hand side, and this portion is called as tDNA. It carries, in the naturally occurring Agrobacterium tumefaciens, it carries genes for plant hormone synthesis and opine synthesis. In addition to these, the TI plasmid has got a group of genes which are responsible for virulence. So what do we call them as virgins? It's a group of genes. They are called as, collectively they are called as virgins. Their gene products are responsible for the successful infection, right? Then in addition to this, it contains a gene, it contains a region which is called as ORIC. So now you know what is meant by ORIC. ORIC is origin of replication. So this plasmid can replicate, okay, along with the bacteria. So there is a specific site or origin of replication, and they also carry genes for opine catabolism. So opines being synthesized by the plant cell will be exclusively utilized by the bacteria for its growth and development, and for that purpose it carries a group of genes which will help catabolize that particular opine being synthesized by the plant cell. Okay, please remember, you have three different types of opine. Not all bacteria will synthesize all three. They will be synthesizing only one type of opine. They will be carrying a gene for that particular opine, and they will be able to catabolize that particular opine synthesized by the plant cell. Okay, so this is a linear arrangement. In a different way it has been shown over here. So you have this left border, as I said, 25 base pair direct repeat. You have a right border, 25 base pair right direct repeat, right? And then you have gene AUXA, AUXB, CYTs. These are the genes for plant hormone synthesis. And then there is a OCS gene. This gene is for opine synthesis. Opine means it can be octopine, it can be nopalin, those three. And please understand, please remember, and I am repeating once again, that these genes have typical eukaryotic expression signals. Now, based on what type of opine it synthesizes, for the sake of understanding what we can do, we can classify these agrobacteria. So you have nopalin plasmids. What does it mean? It means that the TI plasmid carry gene for the synthesis of nopalin in the plant cell and its catabolism, its utilization in the bacteria. Okay. So if it carries a gene for nopalin synthesis and nopalin catabolism, you call it as a nopalin plasmid, and the characteristic is tumor formation will take place, but these tumors can differentiate into shooty masses. They are also called as technically teratomas. This tumor can differentiate and it becomes a shoot-like structure. Now, that is a characteristic of nopalin plasmid. So from morphology also you can say what kind of bacteria must have infected the plant cell by looking at the morphology of the tumor. The tumor formation has differentiated into a shooty mass, shoot-like structure, then one can grossly say that this particular plant must have been infected by a agrobacterium carrying nopalin gene synthesis and catabolism, nopalin plasmid. Second is octopine plasmid. So it is clear now that this particular TI plasmid will carry gene for the synthesis of amino acid octopine in the plant cell, and its catabolism in the bacteria, its utilization in the bacteria. Tumors, characteristically here, tumors do not differentiate. Unlike nopalin, these tumors will not differentiate, but they remain as callus tissue. So there is a minor difference. It is very difficult to really differentiate, but an expert can differentiate, and broadly you can say that the plant must have been infected by octopine plasmid by looking at the morphology of the tumor cell. Third category is agropine plasmid. Okay, so it's clear again that they carry genes for the agropine synthesis in the plant cell and its catabolism in the bacteria. And characteristically here, tumors do not differentiate and they die out. Here, tumors will not differentiate into shooty masses. They will not remain as callus tissue, but over a period of time, these tumors will shed. They will die out and they will shed. So after some time, there won't be any sign that there had been an infection of the agrobacterium, because the tumor must have been removed. It must have fallen down. After some time, it dies out and it falls out, right? So these are the three main, what you can say, based on what kind of gene agrobacterium carries, you can classify those plasmids. Nopalin plasmid, second is octopine plasmid, and the third one is agropine plasmid. Now these opines, as I said, they are used by the bacteria as a source of carbon for their growth and development. Bacteria, agrobacterium utilizes these opines depending on what opine it makes plant cell to synthesize, depending again what type of gene it is carrying, octopine, nopalin or agropine. But whatever opine is being synthesized, that is exclusively utilized by the bacterium for its growth and development. Okay, though it is being synthesized in the plant cell, utilizing plant resources, utilizing the plant machinery, but they will be exclusively utilized by the bacterial cell for their growth and development. In fact, bacteria use it as a source of carbon. Although genes reside on plasmid, but they are active, they are expressed only in the plant cell. Please remember, I have been repeating this all along my lecture, that these genes are there in the TI plasmid of the bacterium, but these genes will get expressed only when the bacterium infects the plant cell, because they contain typical eukaryotic controlling sequences, eukaryotic regulatory sequences or eukaryotic controlling sequences. Therefore, they will be expressed only in the plant cell. So once the bacterium infects the plant cell, these genes will be expressed. Their protein will be synthesized using the resources, the raw material of the plant, using the machinery of the plant. So you understand what is machinery. Machinery, protein synthesis machinery of the plant cell, and resources means the amino acid pool of the plant cell. It will synthesize those amino acids, and then they will be utilized exclusively by the bacteria for its growth. So in this lecture we will stop over here, and we'll continue in tomorrow's lecture beyond this particular point.